Hello and welcome to this presentation entitled Protein Synthesis. My name is Stephen Coffey, and what we're looking at here is an animal cell, and every animal cell undergoes protein synthesis. That's the creation of protein. So an example of that would be bone marrow producing hemoglobin, and there are other protein molecules as well that are all essential for creation and maintenance of life. But before we talk about this important topic, let's just mention the other related topics to it, like DNA and RNA structure, DNA replication, and eukaryotic cell structure. So you need to understand those three topics listed below in order to continue with this video. So what will we learn today? Well, we'll learn the three different types of RNA that are involved in the process of protein synthesis, and also the two main stages in protein synthesis. So these are the key words involved, and they'll be noted in red writing throughout this video, so at any stage you wish you can pause, rewind, and take note of anything you find interesting. So protein synthesis only occurs if you have a certain ingredients in place in the animal cell, and these ingredients are as follows. A supply of amino acids. Now amino acids are floating about in the cytoplasm, and the reason they're important is that they're the building blocks of protein. So proteins are made of joining amino acids into long chains called polypeptides. Instructions are also required as to how to join these amino acids together. That's found in the DNA of the nucleus, and that's the genetic code. So these coding sequences are related to certain amino acids. The type of protein is determined by the order in which the amino acids are joined together. So that's an important thing to note. An assembly line then is required, so it's a protein factory in the cell in the form of ribosomes, and also a messenger to carry the information from DNA to the ribosomes also in the form of RNA. That's what we'll be talking about in the next slide. So there are three forms of RNA. There's mRNA, rRNA, and tRNA. mRNA, or messenger RNA, carries copies of these instructions from the nucleus to other parts of the cell. So what these copies are in the form of codons. Codons are sequences of 3 bases on the mRNA molecule. So each codon is specific to one amino acid, and here's an example of an mRNA molecule. So we've got AUG, a starting codon, and this codon has 3 bases on it, and it's equivalent to one amino acid, which is methionine. So if methionine is linked up by polypeptide chains with glycine, serine, isoleucine, glycine, and alanine, you're going to have a protein as a result of that. The next form of RNA is ribosomal RNA, which makes up a large part of the ribosome. It makes up 40% of the ribosome, and the remainder is made up of 60% protein. So without ribosomal RNA, the ribosome would be structurally unsound and would not be able to produce the protein as it is normally able to use or produce. The next form of RNA is the transfer RNA. This moves amino acids during protein synthesis. So amino acids float around the cytoplasm freely, but when a protein is required to be produced in the ribosome, the transfer RNA collect amino acids and bring them to the ribosome for protein creation. Now the first phase in protein synthesis is known as transcription. So transcription, RNA molecules are produced by copying part of the nucleotide sequence of DNA into a complementary sequence of RNA. So the process requires RNA polymerase. This is an enzyme that's used to bind to the DNA and separate the strands of DNA. So once this enzyme separates the strands, it selects one strand of DNA as a template to assemble nucleotides into a strand of RNA. So how does it know where to start? Well, it starts with the AUG, or starting codon. It always starts with the 3 bases A, U, and G. And it always ends then with one of these 3 stop codons. So UAA, UAG, and UGA. When the whole process is complete, mRNA molecule is released into the cytoplasm and then towards the ribosome. So here's an example of this occurring, or transcription occurring. There's the RNA polymerase. It's unzipping the DNA molecule, while at the same time it's making a template of one strand of DNA, and then it's assembling the nucleotides into a strand of RNA. So once that is complete, the mRNA then it's moved from the nucleus out into the cytoplasm. So the sequence of the nucleotide bases in mRNA molecules serves as instructions for the order in which amino acids join together. So we need something that is able to read these instructions and put them into use. So that's the job of the tiny factory known as the ribosome. So in the ribosome, translation occurs, which is the second phase of protein synthesis. And in translation, the decoding of mRNA sequence into protein occurs. So this only occurs in the ribosome, and during translation the cell uses information from the mRNA to produce proteins. So mRNA, like in this example here, the mRNA, the messenger RNA, attaches to the ribosome. And when it attaches to the ribosome, each codon, or 3 bases, goes through the ribosome, and then the proper amino acid is brought to the ribosome by the tRNA. Once this amino acid is brought and transferred to the ribosome, it's then transferred to the growing polypeptide chain, so the protein chain that's listed here. So each tRNA molecule carries only one kind of amino acid. So the amino acids that are brought here are only relevant to that form of transfer RNA. So in addition to the amino acids, each tRNA molecule has 3 unpaired bases. So these unpaired bases are called anticodons and are complementary to the one RNA codons. So we can see here that ACC is the mRNA codon, and the complementary anticodon on the tRNA is UGG. So that's how the mRNA is able to code what order the amino acids go in to create the protein. So like an assembly line worker who attaches one part to another, the ribosome forms a peptide bond between the amino acids it receives while breaking the bonds that originally held the amino acids to their tRNA molecule. So this releases the tRNA molecule, and also the polypeptide chain continues to grow until the ribosome reaches the stop codon, which is down here. So when it reaches the stop codon, it then releases the newly formed polypeptide, and then the mRNA molecule is released also, completing the process of translation. So when translation is complete, then the protein now undergoes folding, and it folds so much that it reaches a 3D shape, which influences its function. So an example there is hemoglobin, which is produced in the bone marrow. So what we have here now at the end, that completes the process. So if you could answer the questions as best you can, if you need access to the video, you can rewind and pause at different areas. And thanks very much for listening.